And warm welcome also from my side. It is really my pleasure to be the lead-in for Richard to talk a bit about some of our crop science innovation, one in particular that was done thanks to catalytic reactions that have to be cost-effective in a crop science world. And why is that? Why does it matter that we are able to reach with our innovations as many people as possible? Because crop protection helps to sustainably feed the world, because farmers have always been looking for a solution to confront the ever-present threats that jeopardize their crop yields, insects, diseases, viruses, weeds. And in this context, crop science, crop protection has emerged as an essential tool that not only sustains the agricultural productivity, but also plays a role into our ability to feed the world's growing population. You can actually measure this because the net benefit of crop protection is about 30% net average increase in yield for any given crop. So it ranges from 19% in wheat to up to 42% in potato. So that translates to about 550 million tons of additionally produced food crops. And it's an abstract number, but if you translate it into caloric value, this is the amount that could feed 2 billion people. And as we strive to produce better and more efficiently, it is crucial to acknowledge these losses and acknowledge the potential that you can leverage with effective crop protection solutions. I myself am coming from a pharmaceutical background, so it was quite interesting to always compare with the differences that crop protection chemistry with medicinal chemistry has. And striking one is, of course, the contrast in delivery systems, because you can have a pill, an injector, an inhalator, which are all pretty targeted. You have, of course, losses depending on type of delivery, but you are working in a field in the crop science world. And so even though you may be very effective in spraying targeted with drone, on average, the sheer amount of crop protection agents that are needed surpasses a given pharmaceutical compound. But then if we talk back about the commonalities, is that the fundamental principles of developing an active ingredient for crop science are very similar to the ones from a pharmaceutical research, because you always have to target your biological system appropriately, make sure it's safe, and make sure that it's effective. And so as a chemist, looking at the commonalities, it goes further if you think about chemical structures in small molecules. And you can appreciate the sheer variety of chemistry that is utilized in agriculture. And I would want to ask the colleagues in the chat if you know how many of these compounds that are shown here are actually not agrochemicals. Is it 1? Is it 2? Is it 3? I don't know. If you feel like typing it in the chat, I think we can discuss this later as well. Yeah, and I see one colleague got it right. We have here, of course, many crop protection agents, insecticides, herbicides, fungicides, proteoconazole, indaziflam, floxapiprolin, spirotetramat. But we also have one that is odd one out, which is an antibiotic, and that is moxifloxacin on the lower left, which is antibiotic, also by Bayer, known as Avalox, primarily used for the treatment of complicated infections in humans. But you can see a lot of fluorines in the molecules, lots of rings, chiral centers as well. One of the compounds that I would highlight for today is this compound here, which is called bixafen. And this is a great example of how cutting-edge science can capture value for the farmer and everyone involved. So bixafen is sold under the trade name Expro, which is a combination of the names of the two active ingredients in the formulation. It is a combination preparate with bixafen and prothioconazole, so two fungicides, and it has very, very interesting activity not only to keep disease at bay, but also deliver significant physiological effects that are beneficial to the crops. And one of them is, as you can see from the picture, it keeps the plant greener for longer. Which means that this enhanced greening effect promotes increased grain filling. That means that it aids in the accumulation of nutrients in the biomass of the developing grain. And so your overall yield is higher, and a greater number of grains become fully developed and can then be harvested. And it also manages to manage the stress of the plant and allowing for greater allocations towards the grain development. This is so important for high yielding fields. It was launched in 2011. So it's been on the market for quite some time. And if you see trade names like Aviator, X-Pro, Variano, X-Pro, you know there is bixafen inside. It is targeting SDHI mitochondrial complex, succinate dehydrogenase, which is critical to the respiratory chain of funguses, whereas prothioconazole targets the fungal cell wall synthesis. And so when it was invented, lots of different companies were working on this, and Bayer's solution with this mode of action was bixafen. And so let's dive a bit more into the chemistry of how bixafen can be made. So you start with a very widely used reaction, which is a so-called amide coupling, and you dissect the molecule into these two similarly complex building blocks, one of them being this pyrazole 5-membered ring that can then be further disconnected into more readily available smaller molecular weight building blocks. And the biaryl ring is made by joining these two 6-membered aromatic rings together with the reaction, which is named after the scientists that discovered them, Suzuki and Miyaura. So it's a cross coupling, meaning that you couple one ring to another. And we will dive more into this because this will be the focus of the entire lecture today. So let's look into the Suzuki-Miyaura cross-coupling. What is it? It is a reaction that uses a palladium complex as a catalyst to cross-couple an organoboron species, shown in blue, to an organohalide, shown in red. And so this means that we can utilize palladium as a precious metal in order to activate these two compounds here to react with each other and make a carbon-carbon bond. And for this, you use palladium, typically as a salt form, and the ligand, which is an organic molecule that binds the palladium. You can see an X-ray image of a typical palladium-bound phosphine ligand here. And thanks to this agent, these two compounds that would typically not react with each other now have a lower activation barrier to reacting with each other. It cannot be underestimated how important this reaction is for modern chemistry. It was honored in 2010 with the Nobel Prize for palladium catalyzed cross-coupling in organic synthesis at large. And you can see the uptick in literature that references this type of reaction. It's ever growing because it's such an important means of making carbon-carbon bonds. I want to talk a bit more about why this is important from an industrial standpoint. It's an important reaction because it's widely used, because you can make different types of compounds with it. But of course, making a lot of compound with this type of chemistry is not trivial, because palladium is a precious metal and it is controlled by a market that is not the fine chemical or agrochemical market. In fact, most of the palladium used today and for the past 30 years was in automotive application in catalytic converters. There is a small amount of palladium relative to automotive that is used in catalytic catalyst creation, biomedical engineering, so teeth, fillings, electronics, and jewelry also take some part in the use of palladium. But the main use of palladium is not chemistry. And you can also see that the price of palladium, especially in the years where bixafen was on the market, was highly volatile and also started to increase in 2016 before then lowering in 2024. And so it is ever more important to engineer a reaction that can be used for industrial applications, because with bixafen being used for cereals, broad acre crops, you need really hundreds of tons of this to supply an effective solution to the farmers. And so as a chemist, maximizing the yield while minimizing side product formation, minimizing the waste becomes very, very important. In the case of bixafen, even more so, because you can form, not through cross-coupling, but through homocoupling of this dichloroaryl in here, PCB77, so a persistent organic pollutant, which you really don't want to form. And so after many, many rounds of optimizations performed by the colleagues that are cited here at the bottom of the slide, by choosing the right type of organohalide, the right type of boronic acid or boronic acid derivative, and the right type of palladium salt, ligand, solvent, and reaction conditions, you can indeed create a palladium catalyzed cross-coupling reaction, which can be viable on large scale, to the point that 2,000 tons per year of bixafen are produced in Dormagen, so here in central Germany, whereas the Suzuki coupling that you see below is produced in Muttenz on multi-hundred ton scale as well. And this is just to show what can be done with palladium to enable this type of solution to be brought to farmers worldwide. And now I would like to hand over to Richard, that will show you us some more innovative chemistry that you can do with palladium as well. So Richard, if you want to take over, feel free. Thank you.
Can you let me know if you can see my screen?
Yes.
Okay. Thanks so much, Giulio, and thank you all for attending this. Thanks to Tatiana and Hedda and the foundation for giving me this opportunity. I really want to pick up from where Giulio left off. These cross-coupling reactions are extremely important, and Giulio gave us a very nice practical introduction into why that is. And in this second part of the lecture, I hope to give you an impression of what we are doing in my lab at Harvard to continue developing these reactions so that they can be useful and generate products of value to society. Before I start, I just want to say all the research I'm going to show you, all the research that we do in a university, I think is a platform for teaching and training of a future workforce. And I am very, very glad to work with a group of very talented and motivated students and postdocs. I started at Harvard just 3 years ago, just over 3 years ago, and I've already gotten to work with a few dozen very students who have taught me a lot about chemistry and about how to run a group. I'd like to thank our funding sources. I'd like to thank the Foundation for the Early Excellence Award in particular, but all others who have supported our work, they deserve credit for everything I'm about to show you. To give you an introduction into what our group is interested broadly, and then I'll dive into the cross-coupling. One of the areas our group has become interested in is organic redox. So these are organic molecules that can accept or lose electrons. And there's some similarity between those molecules and metals. And so we've really become interested in exploring the reactivity of these redox-active organic molecules, particularly those containing sulfur. So please read about them in some of our papers if you're interested in that area. Another area that I've become very interested in is how to apply organic chemistry to address problems in energy and environment. One of the ways we've contributed is by looking at compounds called photoacids, and recently we've developed the opposite, photobases. These are molecules where if you shine light on them, they become much more acidic or basic. So if you think about basic high school principles of Bronsted acidity and basicity, normally you think of a molecule as having just one fixed acidity or basicity, but there are agents that can reversibly switch between very basic, neutral or very acidic neutral. And these can actually be quite useful in molecular separations, for example, of carbon dioxide. So that's one area where physical organic chemistry and the environment have intersected. But the topic for today is going to be transition metal catalysis. Transition metals, as I'll explain in a moment, are very special elements when it comes to catalysis. But first, I want to start with what they're ultimately applied to do. One of the biggest areas that transition metals are applied as catalysts for is in drug discovery, also in agrochemical discovery, but I'll use drug discovery as an example today. The process of discovering a new drug is still empirical mostly, and it is something that, as many of you know, takes a very long time, a decade or longer. What's the rate limiting step in discovering a drug? Well, there can be several, but if you think about this cycle of design, make, test, analyze, many of us have probably heard about this. I argue that one of the slow steps in this cycle is still the making. So not actually thinking of what compound you want to test next, but the physical process of getting access to those compounds to test is still a limiting step. And it's not just rate limiting, it's, I think, success limiting and creativity limiting. So if you need to make 10,000 to 100,000 compounds to find your final drug, you not only want to make those compounds faster, but you want to make smarter compounds, and you don't want your imagination and your human ingenuity to be limited just by practical considerations of what's makeable and what's not. So that's where synthetic organic chemists come in. We don't actually develop drugs in my group. We develop methods, and then those methods get used by people to make whatever molecules they want. They could be drug candidates, but they could certainly be other functional molecules. We're kind of like the people who provide new shovels for others to dig on the beach for treasure, for example. And that's why I put these little sort of cute shovel emoticons or icons here. What is this? This is an analysis performed in 2016 of what are the most used reactions in medicinal discovery, at least in the publications. What the shovels indicate, those are the only 3 reactions discovered within the prior half century that made it onto this most used list. And those are all cross-coupling reactions. Those are all metal catalyzed cross-coupling reactions. So there is something special about metal catalyzed cross-coupling reactions, that they're sort of the only quote unquote modern reactions that get really, really heavily relied upon in molecular discovery. And we can kind of explain why that is. In general, cross-coupling is first very general, meaning you can put many different kinds of coupling partners in there. And it's also very predictable. I call it one recipe, one cake. If you take one nucleophile, and that could be anything from, let me get my pointer. Yeah, that could be anything from a carbon nucleophile of various kinds to alcohols or amines, things like that. You can take an electrophile, so these are generally aryl, vinyl, sometimes alkyl carbons that are electrophilic. You pick any pair and you get precisely one product out. It's highly predictable and it works a lot of the time. One limitation with cross-coupling, though, is that one recipe, one cake is kind of a double-edged sword. It's also a limitation, because if I wanted to make a different product, what this graph implies is that I'd have to make a different electrophile and a different nucleophile for every new product I want to get. And therefore, if you have, let's say, X nucleophiles and Y electrophiles, the rough amount of chemical space you can access is X times Y. It's sort of a 2D space. So what we wondered in our group when we started was, can you actually develop different reactions, thinking of that as a third axis, where for the same nucleophile electrophile pair, you can actually get multiple related but different products. And that way it's kind of like every time you run a cross-coupling reaction, you could run multiple similar reactions beside it and get 5 products out instead of just 1 or 6 products. And that just accelerates how quickly you can explore different parts of chemical space. So that's as abstract as I'm going to get, but today's talk is going to be about this concept of repurposing cross-coupling intermediates to access as much chemical space as possible. So let's get increasingly more concrete. Let's say we have a pool of electrophiles and a pool of nucleophiles. Conventional cross-coupling will just take one electrophile and one nucleophile and link them together. So there's different electrophiles you can use. There's a few different examples of nucleophiles you can use. People tend to name the reactions after what nucleophile is being used. And in today, I'd like to cover two different topics of investigation in our group. One question could simply be framed this way. Instead of connecting those two electrophile and nucleophile together, could we insert a small third component? That idea on its own is not entirely new, but hasn't really been as systematically explored as one might suspect, and I will talk about that in a moment. In the second half of the talk, I'd like to talk about a concept we call migratory cross-coupling, where you don't make the bond where the activating group was, but the metal somehow is able to walk to a different position and make what we call an isomeric product. So can you actually get catalyst control over different isomers of product and not have it just dictated by the starting material? So we call that migratory cross-coupling because the metal participates in a migration. So let's start with this idea of third component insertion. Why might someone want to do that? So this is a preclinical candidate that was published in the literature. It's a pretty standard structure. It's you can see a CC bond here that's probably made by Suzuki coupling. And then this linkage between these two aryl rings happens to be a diaryl ketone. And the point of this slide is that it wasn't a diaryl ketone to begin with. As they detailed how this study proceeded, they considered many different ways of linking that pyridine and that arine, either directly with a CC bond or with a nitrogen in the middle, a substituted nitrogen, an oxygen, a carbonyl, an alcohol, etc. And to analyze a little bit further, how did they get to all of these compounds? Well, for every single one, they had to get a different pyridine starting material and perform a cross-coupling. For example, for this ketone, they could do nucleophilic addition to a nitrile and then hydrolysis. This actually is totally fine if the group over here is pyridine, because you can buy all of these things. It might be annoying to have to buy them, but it's totally feasible. Now imagine this was a parallel campaign where this group wasn't pyridine, but was something substantially more complex. This poses more of a challenge because then you would have to go and remake or convert these functional groups from one to another at a late stage in order to access 5 different nucleophiles, to access later 5 different compounds to test, or 6, however many. So here's our vision. If we could instead just have 2 coupling partners, let's say the Suzuki pair of an aryl bromide and an aryl boronic acid, and then add reagents that are the equivalent of nitrogen, oxygen, a carbene, for example. And here we have the insertion of carbon monoxide. That happens to be a known reaction, so this one is actually possible already. But the others, surprisingly to us, hadn't really been systematically considered. How do you make all of these different fragments by insertive couplings from the same pair of starting materials? Okay, so to get even more concrete, Giulio just gave us a very, very nice primer on the Suzuki Mura coupling. Probably the second most important class of couplings in medicinal chemistry is called Buchwald-Hartwig. It's the carbon-nitrogen coupling between an aryl halide and actually the nitrogen atom of an aniline or amine. So when we started, we were just thinking, well, this nitrogen insertion idea is effectively linking the Suzuki coupling pool to nitrogen containing products. So this is a project about how we can repurpose Suzuki coupling partners, which many companies have tens or hundreds of thousands of them, and make diarylamines, which are important pharmacophores directly. Okay, so this sounds, why is there a talk about this topic? It sounds pretty simple. You just couple A, B, and C. Turns out the more components you have in a reaction, the harder it becomes to realize systematically. And here's why. What we're trying to make is a big blue ball, big red ball, and a small blue ball in the middle. This is a nitrogen and NH, let's say. Well, what is that reagent that goes in the middle? It's going to have to react both with the nucleophilic side and the electrophilic side. So it's going to be something that we call an ambiphile, something that's both has nucleophilic and electrophilic character. If you enumerate the kinds of organic compounds that have that character, they tend to be very reactive things. Carbene equivalents, in this case, nitrine equivalents, things like that. And if you have a nitrine equivalent and low valent palladium, the first thing you have to worry about is that those could just react directly and lead to something off cycle. that won't do catalysis. So let's suppose we're able to avoid that somehow, perhaps by making this blue reagent less reactive, and we can undergo the first standard step of cross-coupling, oxidative addition into the electrophile. Then we need, we're trying to make A, B, C. We don't want A and C to couple directly, the ends to couple directly. That turns out to be one of the hardest challenges because Suzuki coupling, direct Suzuki coupling, is one of the most well-established and optimized reactions and proceeds so quickly for many different kinds of palladium catalysts. So how do you slow that down long enough for this middle reagent to then insert? And then assume you can insert that middle reagent, bring on the C, the other end, the nucleophilic reagent, and you make something like this. Once we get here, I think we're pretty confident in the final step because that's in common with Buchwald-Hartwig coupling or standard cross-coupling. That's just reductive elimination. So here's a plausible sequence if you could do all those things to make A, B, C. And then on top of that, you have to worry about selectivity. We don't want to make A, B, A or C, B, C or A, B, B, C. So as you can see, there's a combinatorial challenge when you increase the number of components that go into a cross-coupling. So long story short, here's the first reaction we were able to discover and report. This is a standard Suzuki coupling pair. And then in the middle, we have this ambiphilic reagent. It's a nucleophilic because of the nitrogen lone pair. It's also electrophilic because of this NO bond. And when you mix these components together with a standard a standard cross-coupling catalyst, you can get either the ABC 3-component product that we want, or you can get a lot of times the direct coupling product, which we don't want. So that's going to be in the light blue. And you can see with some ligands like Brettphos or Rufos, you get quite a lot of this direct coupling product. And it's only when you go to these ligands that have T-butyl groups on the phosphorus that you start to shut that down and get a lot of the desired product. The reason for that is these groups are very big and they kind of prevent that transmetallation step for long enough that the nitrogen reagent can really insert. So using very large bulky ligands is going to be one theme of this talk. Some technical details, you can use potassium phosphate as our standard base. The 4 equivalents is actually very important. If you just go down to 3.5 equivalents, the reaction doesn't work as well. Potassium hydroxide is another effective base. And other reagents of this type don't tend to work as well. This phosphinate leaving groups tends to be quite important. If you want to use the pinnacle ester instead of the boronic acid, which is very popular alternative, just use potassium hydroxide and the reaction works fine. I don't like to belabor scope. I just want to show that this reaction is very, very general in our hands using the aryl triflates. Pretty much any electronic substitution, electron-rich, electron-poor groups, those are all tolerated. We can, the conditions are very mildly basic, so they tolerate a sort of epimerizable stereocenters, Bach groups. Aryl triflates react over aryl chlorides, so that can be useful. Alcohols are tolerated, albeit in lower yield. Some heterocycles as well, including pyridines, quinolines, thiophenes. And for these kinds of challenging boronic acids, recommend using the pinnacle ester and the potassium hydroxide prep. That tends to work a lot better. And this is a reaction with an aryl tosylate in case you're interested in using those. Reaction works very well with aryl bromides and chlorides as well. We didn't do as many examples, but hopefully this makes the point effectively. Here's an interesting example. This is actually a recently approved drug that was made by Suzuki coupling at this position. You can just imagine, we just made that drug. We have these 2 intermediates already. We don't need to buy anything new or prepare anything new. Just by running the same reaction, inserting this reagent, you can now get the effectively nitrogen inserted analog, which you may want to test. Okay, so we talked about making ABC. What is exactly the mechanism for doing that? The whole story would be way too complex. I just want to focus on one question, which is very natural. If you're making ABC, you clearly must be making AB first or BC first. One of those bonds has to come first. And therefore, we can divide the potential mechanisms into 2 classes. The so-called electrophile first mechanism, which I'll explain now, and the nucleophile first mechanism, which I'll explain in a moment. In the electrophile first mechanism, palladium undergoes oxidative addition to your electrophile, and then this amine reagent will coordinate. When it coordinates the palladium, this interesting process can happen where the aryl group migrates from the palladium to the nitrogen, and this phosphonate group then leaves and then ends up back on the palladium. So this is a net diatropic rearrangement, but the key part that I want to highlight is we've made the electrophile nitrogen bond first. After this, this phosphonate group can undergo transmetallation with an aryl boronic acid, and then straightforward reductive elimination leads to the product. Okay, so one thing we can do in 3-component couplings that you wouldn't want to do in a 2-component coupling is leave out one of the components. If we think the electrophile can react first, well, we can actually just make a species with the electrophilic aryl on the palladium, react it with the amine reagent, but don't add any nucleophile, just to see if there's a possibility of making a CN bond between this red aryl and this nitrogen. Indeed, we can. You might note the 3% is quite low, but we also end up with 25% of this product, where this aniline, because there's no other suitable nucleophile, goes back into the cycle and couples again with another aryl group. So both of these are indicative of what we were trying to test, which is, can the electrophilic aryl group make a bond to nitrogen under our conditions? It certainly can. In the nucleophile first mechanism, that also begins with oxidative addition, but now both the nucleophile and the aminating reagent coordinate in this ternary kind of complex. Why would we invoke such a complicated species? The reason is because in the nucleophile first mechanism, it's the nucleophilic blue aryl group that has to migrate to nitrogen first. And indeed, that can happen through this intermediate, which after hydrolysis of the boron nitrogen bond leads to this familiar intermediate. This is just like what was at the 9 o'clock of the previous cycle, except the nitrogens on the other aryl group. Nevertheless, reductive elimination is again known to be straightforward here. And in this case, it's a little bit harder to test, but what we can do is we can mix these components and vary the amount of palladium we're adding into the reaction. For certain substrates, like this pair, you can actually start to form carbon-nitrogen bonds between the blue aryl group and the nitrogen, even without palladium. But as you add palladium, that forms more and more, and then furthermore, some of this aniline that's formed can go and do cross-coupling with this red aryl group to make what is our desired product. A little bit complicated of a slide. If there's one takeaway, it is that there can definitely be coupling between the blue aryl group, the nucleophile, and the amine reagent under our conditions. Okay, and because that can happen either with or without palladium, there's this parallel pathway that we proposed and later studied, where this boronic acid is in situ converted to an aniline, and that undergoes Buchholz-Hartwig coupling. That operates in parallel with that palladium involved mechanism that I described earlier. So in both of these ways, the nucleophile first mechanism also seems to be viable. And you know, to cut a very long story short, what we truly believe in these reactions is that there are both mechanisms, both the electrophile first and nucleophile first mechanisms operating simultaneously, or one operates for some substrates and the other operates for other substrates. And remarkably, even though both are operating, we never get products like ABA or CBC that we were worried about previously. Okay, so the fact that multiple mechanisms operate is actually an advantage for extending your reaction, because you can introduce new steps or new components into either of the mechanisms, and therefore there are multiple viable ways for that mechanism to grow into something more complex. For example, we can do these 4-component couplings where we formally inserted both CO and NH, and we can actually now do that in both orders. Specifically, we can do allylation reactions where the allyl serves as the electrophile, and you get the same isomer whether you start from the linear or branched isomer of the electrophile. So these are examples where we're extending that nucleophile first mechanism. There are examples where we can extend the electrophile first mechanism. We know that we're extending the electrophile first mechanism because when you use alkyl boronic acids, we observe a lot of this diarylamine, this homodiarylamine from the electrophile side. So it's clearly what must be going on in this reaction is CN bond formation with the electrophile. And so how do we get that product exactly? This is the cycle I had about 10 slides ago. When you get to here, remember what we needed to do was to do transmetallation with the aryl boron. Well, now we're looking at alkyl borons. So alkyl boron does not transmetallate as quickly as aryl boron, meaning the step right before, the intermediate right before, has time to do something bad. That something bad is hydrolysis to make free aniline. And when you have free aniline, it can enter the cycle at this 2 o'clock position and make that undesired homodieryl coupling product. So the challenge of this project was to shut that down. A very good graduate student, Sophia, was able to do that. And now we are able to use primary and secondary alkyl boron species in these aminative couplings. Here's just a few examples. I like this example. For example, you can imagine you could make this by hydroboration of the allyl ether, followed by this aminative coupling to get a pretty complex drug-like structure. Okay, this method is also, because of this mild base, sodium silanoate, it does actually tolerate some base-sensitive groups, for example, another protected amino acid or the sulfonamide. We did extend this method also to oxygen atom insertion using this reagent and methylmorpholine and oxide. It is able to formally insert an oxygen atom in between sort of a Suzuki coupling. And this is sort of an interesting analysis we did. I'll explain it to you step by step. We took a catalog from a large chemical supplier, and we tried to analyze what kinds of phenols are available and what kinds of boronic acids are available. Phenols, are shown in blue, tend to concentrate in certain areas where there's not a lot of red. And then the aryl borons tend to concentrate in other areas where there's not a lot of blue. So if you're able to use aryl boronic acids as surrogates for phenols, you can actually access a totally different set of products and explore a larger swath of chemical space. Put it this way, if you were doing some CO couplings, you would want to access as much of this space as possible, not just where the blue coverage is, but the union of the blue and the red is clearly kind of a larger and more attractive area of space to explore. This reaction works interestingly very well and with a scope sometimes greater than what you can do if you just put in the phenol. And here's how it works with aryl bromides, chlorides, and triphlates, and here's how we believe it works. In our mechanistic studies, if you mix, here we're using a pinnacle ester just so we can track these things by boron NMR. If you mix that with the oxidant, what you get immediately is actually not phenol, but sort of this borate species where the oxygen has inserted, but this is not an H. It's there's no source of H+ in the reaction. It's just bound to boron. It actually wasn't clear to us that this species could transmetallate the way we needed it to after oxidative addition with this oxidative addition complex. But indeed it does, and it does that very effectively only when you add a little bit of organic base. So that was the key finding in these reactions. So I'm going to leave the insertion chemistry here. We have gone on to insert other things such as carbon and do more complex 4-component and 5-component couplings. Hope to tell you about that in a future lecture, but I do want to spend some time on the migratory coupling with our remaining minutes. This migratory coupling really comes from an observation that wasn't made by us, but was made by the Buchwald group between 15 and 10 to 15 years ago. They were trying to do a very difficult coupling, carbon-fluorine coupling. And these are difficult because fluorine is very small and hesitant to reductively eliminate. You know, something I truly believe is when you do difficult reactions, try to do difficult reactions, that's when you start to find very interesting unexpected chemistry, because the thing you're trying to do is very difficult, and that gives the molecules an incentive to do something else. That something else in this case was to generate a CF bond, but at the quote-unquote wrong position. And this would happen for a few different substrates. The way that happened is through formation of a palladium arine. And so you can look at this diagonal arrow first, because that's what they were trying to do. If fluoride could come substitute this X at the palladium, and then you did reductive elimination, that's where we want to go. But that fluoride could also, because it's basic, deprotonate the ortho hydrogen here, making this palladium stabilized benzyne intermediate. And if the HF that just left, if that comes back on exactly the way it left, you can actually just get to the species you want. But there's another option, which is HF can come back the quote-unquote wrong way. It could come back from the other end of the benzyne. And then effectively what we've done is swapped the positions of the palladium and the hydrogen. And from here, reductive elimination would give you the meta product. So quite a lot of effort was actually put into shutting down this left mechanism because a mixture of fluorides is not something that's very easy to separate nor desirable. When we started on this project, we asked ourselves a very simple and kind of humorous question: what if we on purpose tried to make the wrong product and not just do CF coupling, but do other kinds of useful cross-coupling reactions, making quote unquote the wrong isomer on purpose? So it's a kind of a contrarian motivation to starting picking up this work 10 years after the Buchwald studies. I will skip the optimization, but we found that with a very hindered ligand, so we've gone even to adamantyl, not tert-butyl. Adamantyl is even larger, and protic solvent, which is key. The role of the protic solvent is to prevent CF reductive elimination. And if you prevent it completely, what you see is actually a very funny reaction, which is just chloride walking. So what's happened here is the palladium's inserted in there, walked to the next position, and then just reductively eliminated chloride. And depending on the group here, the equilibrium ratio that you reach can be anywhere from 0.2 to about 3.5. And we know it's an equilibrium because you can start with either meta or para N-acetyl chloride, and you can converge the same ratio of isomers. So this kind of chloride walking is very interesting. Are you doomed to get then a very bad ratio of products because this is thermodynamically controlled? Well, we thought possibly not, because let's suppose these chlorides are interconverting somehow, and we couple that with a reaction where some nucleophile can come in and intercept the two different chlorides. Well, in this scheme that I've drawn, it's hopeless, because the barrier here is so high that you're in this kinetic quench regime where the ratio of these chlorides has a big influence on the ratio of the products that you get. But as you lower the concentration of nucleophile, you effectively make these couplings on the sides harder and harder. And at some point, they have a higher barrier than that isomerization process. And that's a characteristic of Kern-Hammett control. So we're letting the molecule equilibrate many, many, many times, the chlorides equilibrate many, many times before engaging them with a nucleophile. And what that allows is for the selectivity, therefore, of the coupling step to dictate the product distribution rather than the intrinsic thermodynamic ratio of the chloride. So this is, you can think of this as dynamic kinetic resolution, not of stereoisomers, but actually of constitutional isomers. So we were able to do that with oxygen electrophiles. You can see in some cases you can get ratios of about 10 to 1 or even higher in favor of the meta product, which is the harder product to get in these cases. And the scope is actually quite general. It even works with alkyl, with sort of aliphatic alcohols, both primary and a few secondary, and we are able to do nitrogen nucleophiles as well, mostly weaker nitrogen nucleophiles such as anilines and sulfonamides. So these are, these yields could be improved, but certainly from a medicinal chemistry standpoint, to get a little bit of a second isomer essentially for free could be advantageous in some cases. Just to show you the reaction mechanism in more detail, what's going on is we have the formation of a palladium fluoride complex. That fluoride, actually, when you have very large ligands here, that fluoride gets pushed very close to this ortho H, and that's able to essentially eliminate that HF and make the palladium benzyne through this sort of very beautiful butterfly-like transition state that we calculated. That benzyne then needs to rotate, flip around, so that the HF can come from the direction it just left. And this benzyne rotation is something I'm happy to talk a little bit more about. It's more important than one might think, but assuming you do rotate and the HF comes back, then you've essentially walked your palladium, and that's how we believe this happens, every single step being reversible. Some deuterium labeling experiments, so if you use deuterated solvent, are consistent with this, with deuterium washing into eventually all of the positions as the palladium walks around the ring. Okay, so I just want to show you two quick applications before I close. This is actually an insecticide that's made by CO coupling. Conventionally, you would just get the oxygen essentially where that bromide is. But using our migratory coupling, you can actually get it to the meta position. Now, let's say you took pyroproxyfen and you wanted to brominate it. You can't meta-brominate it, you can only para-brominate it. So then we could still get the meta product by using our migratory coupling. Thought this was an interesting demonstration of one case where you might think about using our reaction. Here's also a pharmaceutical derivative that we were able to couple this sort of peg-like chain onto it. So I've shown you two reactions here. I'm going to skip ahead a bit. Sorry, past this part. So in summary, I've shown you these two reactions today. Sorry for that skipping. I've shown you a set of reactions where we insert small third components into cross couplings that we're looking very forward very much to developing further. And we also introduced the concept of these migratory cross coupling reactions, which I think provide very fertile ground for the development of, for example, migrating further, migrating to and from the ortho position, migrating to other rings, etc. And we look forward also to exploring those. One piece of work that I did not have time to talk about today, but I hope you'll read about, are these formal sulfur deletion reactions. These are reactions where we make carbon-sulfur bonds by nucleophilic aromatic substitution, by SNAR or by cross-coupling. And then we've developed nickel catalyze methods to essentially remove that sulfur atom but reconnect the ends. And if you read that paper, it will first explain how we did it, but also give you some sense for why we're excited about these deletion reactions and what they could actually be useful for. In other words, why would you ever introduce a sulfur only to delete it later? And we hope we've made some case for that in that paper. So I just want to provide you with the reference there. Seeing as our time is almost up, I just want to say thank you all for attending this talk. I want to thank again my group. Those highlighted in italics are those who did all the work that you saw today, but I owe my entire group tremendously for the opportunity to do this science and to continue it over the coming years. So with that, I think maybe I will just leave it here and answer any questions that you might have.